Welcome back, children. Now, in the previous videos, we are discussing about different types of cells, sizes of cells, shapes of cells, and basing on the cell present in their body. If a body is composed of one single cell, we have seen that it is a unicellular organism. And if a body is composed of more number of cells, it is called as multicellular organism. And in the previous video, I have clearly explained you how a unicellular organism is able to maintain all its functions. Now, this single cell is capable of undergoing all the functions necessary for the body. Maybe respiration, maybe excretion, maybe circulation, whatever it is, for an organism to exist, whatever functions are necessary, all the functions are undergone only by that single unicellular. That means that single cell will undergo all these functions. So that is why it is called as what? Unicellular organism. Uni means one. Uni means one. So unicellular means a single cellular organism, and multicellular organisms are designed with more number of cells, and they are able to function like respiration, excretion, circulatory system, whatever it is. Every cell has its own peculiar function. Like that, these organisms are able to manage with unicellular and multicellular. Now let us start the activities present in this lesson. Now, as you are very new to this biology, so let me start with observing of matchstick. Now, in the first lesson itself, in the first video itself, I have mentioned you that each and every organism is made up of cell. Maybe a bird, maybe an animal, maybe a plant, maybe a organism. Whatever it is, every organism is made up of what? Cell. Every organism is made up of cell. Now, if you want to observe how the cells are, and already I have mentioned you that cells are very, very small, that they can be observed only under microscope. So, the device used for observing the cells is what? Microscope. Now, let us take the material called matchstick. What is that? Matchstick. You all know matchstick, no? So, first of all, we are doing an experiment on matchstick. Okay, so the material what we are selecting is called as what? Experimental material. So, like that, if you observe Robert Brown discovered what? Nucleus. So, Robert Brown took which experimental material? Orchid leaf. So, the experimental material selected by Robert Brown is orchid leaf. Likewise, if you take Robert Hooke, he is the person who discovered cell. The experimental material selected by Robert Hooke is what? Oak tree. Like that, for you to observe cells, we are selecting an experimental material called matchstick. What is that? Matchstick. So, how to observe matchstick cells? So, by this you can understand that each and every organism is made up of cell. Now, matchstick is a part of what? Part of plant. Every matchstick if you observe, this matchstick will be prepared from what? Plants. Now, first of all, take a matchstick. First of all, take a matchstick. And you soak that matchstick in water for half an hour. We have to soak that matchstick in water for how many hours? Only for half an hour. Okay, as it is not for burning, you need not worry. Even though you soak matchstick, nothing will happen here because you are not using it for lighting. Just you are using it as for experimental material for observation of cells present in the matchstick. Now, take a matchstick. You take a bowl of water. In that water, you just keep this matchstick for about half an hour. Now, after half an hour, take out that matchstick and cut it into thin slices. A sharp blade will be present in the laboratory. You take it and cut it into a very thin slice. Now, after cutting it into a thin slice, take a very thin slice. So, for your convenience, this is slide now. What is this? Slide which is used under microscope. This is called glass slide. What is this? is called glass slide. Now what we are going to do here is, we have to cut this matchstick into very thin slice. Now select one of the slice. Select one of the slice. Now we have to place the thin slice on the slide. Slice, slide. Don't get confused. First of all, what did we do? We took a matchstick. We soaked it in water. Second step. After half an hour, take out that matchstick. Take a sharp blade. Make it into thin slices. As if like potato slices how you make for preparing Lay's packets and all. Right? Like that you make a very thin slice from that matchstick. And now what you have to do is, you have to place it on the slide. This is called slide, which is used under microscope to observe the cells under microscope. Right? Now take that one of the thin slice and place it on the slide. Place it on the slide. Place it on the slide. Okay? Now you can take a drop of water. You can take a drop of water, or else you can take a drop of glycerin. Why miss? Why to add drop of water, or why to add a drop of glycerin? Why? Because it won't allow the slide to get dry. The experimental material won't get dry when you use glycerin or water. So what will happen here is, when you place this experimental material, the thin slice on the slide, put a drop of water, and now you're going to cover it with a cover slip. You're going to cover it with what? Cover slip. And now you can observe this under microscope. You can observe this under microscope. So you can find structures like this. You can find the exact structure will be like this only. The structures, these are called cells in the matchstick. Cells in the matchstick. Okay? So actually these are dead cells. The cells, what are dead cells mean? The cells which are not functioning, which are incapable of functioning, is called as what? Dead cell. The cell which can undergo function is called as living cell. Okay, ma'am? Now the cells which can undergo function is called as a living cell. Which cannot undergo function, non-functional cells, are called as what? Are called as dead cells. Now these are dead cells, and you can clearly observe. Now, can we observe living cells also? Can we observe living cells also? Take an onion peel now. Take an onion peel. Peel of an onion. Cut it into thin pieces. Cut out a small portion. A thin membrane will come out from that onion. A thin membrane. You can find very clearly. If you want, after this video is finished, you can go to your kitchen and you can find out. A thin membrane will be present on the onion. Now, you just take a very small portion from the transparent membrane and place it on the slide. Place it on the slide. Same process again. Cover it with cover slip. Observe it under microscope. Observe it under microscope. You can find cells like this. These are called living cells, and these are the cells present in the onion. Present in the onion. Present in the onion. So these cells are present. These are the cells present in the matchstick. These cells are present in the onion. Now if you want to observe, you already know that in the year 1831, which year, ma? 1831. Robert Brown is the scientist who came forward to explain you the clear structure of cells with the nucleus. Now if you consider both matchstick, these are the cells in the matchstick. Now if you observe onion peel, these are the cells in the onion. Now both these are examples from plant kingdom only. Now can you observe your cells also? If you take blood, you can observe blood cells. If you take the scraping from your cheek, the inner lining of the cheek, you can observe cheek cells. You can observe which cells? Cheek cells. For that, what you are going to do is take an ice cream spoon. Ice cream spoon. Now, from the inner lining of your cheek, that means you should not touch this side. You should not touch this side. You should take the inner lining. Okay, because mucosa membrane will be present, which you will be studying in your 9th and 10th classes, of course. You need not worry about that. Now, right? Now, what you have to do is, you have to connect the scraping of your cheek from the inner lining of the cheek. Now, you take that and keep it on the watch glass. Keep it on the glass. And now, transfer it to the slide like this. Collect the scraping, collect the inner mucosa membrane of which cells? Cheek cells. Now, place it on the slide. Place it on the slide. In the same way, cover it with cover slip. Cover it with cover slip and observe it under microscope. You can observe cheek cells. You can observe cheek cells. Now, if you want to observe nucleus, now what am I saying you is, here all the things what you are observing is simply you are observing cells. But you want to focus on nucleus because you are familiar that in 1831, Robert Brown is the scientist who came forward with an intention to find out what is internally present in the cell, and he found out that every cell is possessing a special peculiar body in the midst of the cell called nucleus. Okay, and now to observe nucleus, what we can do is, you can observe nucleus in the onion peel. First of all, after selecting the thin slice, you cover the thin slice with a stain. What do you mean by stain? Any color. You take saffron, you take red ink, you take blue ink, you take iodine. Any staining body you can take. Okay? You can take methylene blue. Anything else you can take. If nothing is available, you can take your red ink or blue ink also. Now, after placing of that mucosa, or else you can take onion peel, because even the plant cell is also having nucleus, and also animal cell also is having nucleus. Any of these you can select. Okay, and now place it on the slide and put a drop of crystal violet or methylene blue or else saffron or else red ink or else blue ink, anyone else you can take, and put a drop on that material what you have taken. And wait for a couple of minutes in the sense 2 minutes, or else you can wait for 5 minutes. You can wait 2 to 5 minutes. After that, just take a tissue paper or a blotting paper and remove the stain, excess stain, whatever it is. You just remove it and again cover it with cover slip. And now observe it under the microscope. You can observe cells with a dark spot in the middle. You understood the difference here? Simply we have done the observation of cells here, whereas if you observe here, we have stained it. Because of that staining, if you take blue color, everything will be blue, but the midst of the cell will be black spot like this, and this black spot indicates it is nucleus. It is what? Nucleus. So by this we can understand that cells are possessing a dark staining body in it, a special speckled body in it, which controls the functioning of the whole cells in the body. That's why it is called as cells control room. It is also called as manager of the cell. It is also called as the leader of the cell, which manages the functioning of the whole cell. So in the three main parts, what are the three main parts you studied? Cytoplasm, cell membrane, nucleus. Who is the leader here? Nucleus. Because it controls the functions of each and every function of the cell. Okay, now this is the way how to observe nucleus in the cells. If you stain, you can observe nucleus. If you don't stain, if you just use a drop of water, normally you can observe cells. Normally you can observe cells. Okay, so in this way you have observed matchstick. In this way you have observed onion peel. In the same way you observe nucleus, and also the cheek cells or else the onion peel. Any cell if you take, they are having a dark staining body inside it. How to observe it? By staining it. What do you mean by staining? After taking experimental material, if you use any staining in the sense any color, that is called staining. After waiting for 2 or 3 minutes, you have to remove the excess stain and cover it with cover slip and observe under a microscope. You can find a dark staining body in the midst of the cell. The midst of the cell, that is called as nucleus. So these are all the activities which are present in your second lesson. Okay, so in the next lesson we are going to end this chapter now. So keep watching. Meet you back in the next video.